Hello again, Biology 300 students. Mr. Parker here, and in today's screencast, we're going to be talking about protein synthesis. This will actually be one of 3 screencasts for proteins. The second one will be on the topic of mutations, and then the third one would be on enzymes. Okay, so you can see here a little picture of what's going to be, what we're going to be talking about on the screen here. But before we kind of start talking about that, let's go ahead and go over the learning targets. As you can see here, that we are going to be able to answer as we go through and as we finish this up. And then the textbook reference is chapter 10.1, and then the packet that you should have out is your protein synthesis packet, DNA and protein synthesis, and the pages we'll be looking at are 91, 92, 93, and then also 95. So you should have those handy. And then you should also have your screencast note-taking or screencast learning targets out, so you can go ahead and answer those and write down your notes as you follow along, so you have all your information organized. All right. But before we kind of get into the whole idea, well, it's not before, but as we go into the whole idea of protein synthesis, what we really need to look at first is the structure, the actual structure of RNA. Okay, and looking at the differences between, as you can see up here, DNA and RNA. Let's look at the nucleotides first. Okay, so the nucleotides we know are all made up of phosphates. They're made up of this 5-carbon sugar here, and then they're also made up of the nitrogen base. And when you look at the 5-carbon sugar, the major difference between the two here, you can see is that we have one has the OH and one does not. So RNA is referred to as ribose sugar, and whereas the DNA is referred to as the deoxy because it's missing the oxygen. When we go into looking at the actual nitrogen bases, DNA, the only difference is that DNA has the thymine, whereas RNA has the uracil. And the bonding is the same. A goes with U, G goes with C. So when we're talking about RNA, we never have the molecule here of thymine. And then looking at the structural difference, the third difference here, when we're looking at the polynucleotide chain, you can see here this is the double helix. You got the 2 sides here to the ladder, and then whereas RNA is the single, and it's able to move throughout through the cell membrane and go off into the cytoplasm, eventually to the ribosomes to make the proteins. All right. So this RNA is necessary to carry that genetic information from inside the nucleus, outside the nucleus, till we need to go off and make the ribosomes. All right. So looking at some of the different types of RNA, there's actual 4 different types that we'll actually make a little bit. We'll take a closer look at 2 of them. But there is 4 total types here. You have tRNA, messenger RNA, rRNA, and snRNA. With the messenger RNA, basically what that does is it encodes for the amino acid sequence of the polypeptide. And what I mean by a polypeptide, I'm talking about proteins. That's what messenger RNA is going to do. It's codes for the protein. And where does it get the code from? The code comes from the original template, which was the DNA. We'll talk more specifics about that here in a few minutes. Then we have the transfer RNA, and that's going to bring the amino acids to the ribosomes, and this is during the process of translation, and this is going to occur off into the ribosomes and the cytoplasm where translation occurs. So your tRNA again plays an important role. It's called the anti-codon. Okay, and then you can kind of see the other 2 here. I'm not going to go through those too much, but you can kind of see what their role is in the process. Now, looking at a little bit more about the messenger RNA. All right, the messenger RNA here carries the actual genetic code that specifies for the amino acid sequence in the polypeptide. And as I mentioned in the previous screen, the polypeptide is what we're talking about as a protein. And proteins are made up of amino acids, and we'll take a look at the building up of the amino acid here in a few minutes. So as we move into the process of converting or transcribing from the idea of DNA into RNA. And remember, we're working with the same idea or the same language, nucleic acid language. We're going from DNA to RNA, but we have to change the code a little bit so it can go off and go into the cytoplasm. So where it all begins is with the RNA polymerase. The RNA polymerase is going to bond to this promoter, okay, which is the region of the base that signals the beginning of the gene. So this is going to say, all right, The gene is here. We're going to start reading it, and we're going to start coding for RNA from our DNA template. And the double helix will unwind at this point and is ready to be transcribed. So we're not really changing the language, but we are transcribing it from DNA to RNA. And that will allow us to eventually leave the nucleus and go off and do the job of where the ribosome is going to do what it needs to do. So moving on to the second step then would be elongation. And this is kind of very similar to DNA replication, where we had to elongate or make more or put more nucleotides in. And as you can see in the animation, what's going to happen is this the RNA polymerase moves along the protein, and we're going to add in more of these nucleotides. All right, so what the RNA polymerase is doing, it's moving along and adding in the nucleotides. Now remember, in DNA replication, there was something called DNA polymerase, and what was its job? It was to add and proofread. Okay, well, same thing with RNA polymerase. What it's supposed to do is add and proofread from the RNA, and it uses the DNA as the template to be able to make its own strand. And you can see here, here's your RNA strand that's been formed. This green structure again was your RNA polymerase that's moving along and coding for the new substance. And these nucleotides are added about 6 nucleotides per second. Okay, moving fairly quickly, and then eventually what's going to happen, the messenger RNA leaves and it will go off and do the next step. So the last step here is the termination. This is when we get to the end of the coding gene, and the RNA polymerase reaches this point, and basically it's going to stop the process of transcription, and the enzymes and other factors will help release it. And we're going to then basically end the process of transcription here. So you have step 1 was the initiation with the RNA polymerase, unwinding it. Step 2 was the elongation, where we're adding more nucleotides in for the RNA. And step 3 is the termination process, where the process of transcribing the RNA is going to be stopped. And eventually, as you see here, the messenger RNA is going to be released off into cytoplasm. Okay, so moving on from transcription that occurs within the nucleus, we're going to be moving outside the nucleus, and you can see that in the little picture here, a little diagram. All right, what we're doing is we're transcribing from DNA to RNA. Now we're going to go through the process of what we call translation, and this is where we're making these polypeptides. Now remember, again, polypeptide is a protein, so that's what we're making are these proteins, and this is occurring out in the cytoplasm. Okay, and it occurs right at the ribosomes. Okay, we have this DNA, which is an RNA, which is nucleic acid language, and they use a 4-letter basically language. And you're talking about A's, U's, G's, and C's, right? When we're talking about RNA. Now, when we're moving into proteins actually use a 20-letter language, and that language would be the amino acids. There's 20 total amino acids that has been coded for by the RNA that's originally come from the DNA template. So what you're going to see here is what we refer to as the codon chart, and this codon chart is you can kind of see, these are the amino acids. These are abbreviations of amino acids. As if you went through them and counted them up, there's 20 total. And you can see there's different combinations of our nitrogen bases, depending on what order they're in, that will code for a certain amino acid. And depending on the length of our protein structure and the shape of our protein structure determines on what its function is. And I'll go over a little bit on how to use these codon charts here a little bit later, and but it's the messenger RNA that we're going to be using to help us determine what the protein structure is, putting together our amino acids. Okay, so what are the major players in this process? There's 4 major players in translation. There's messenger RNA, there's ribosomes, there's transfer RNA, and then the amino acids. And we're going to go through each one of these real quick, kind of give you a little bit more background on them, what their role is, and their importance in the process. So first off is the messenger RNA. And looking at this, what you're going to see here, or what you see here, is this is a long strand of RNA. okay, the messenger RNA. And you can see the individual nitrogen bases that are located on here. And there is something that you can see up top here that's referred to as a codon. And on the bottom it's defined as, basically your codons are a 3-base sequence of messenger RNA. So in this particular case we're talking about AUG, that is a codon. In this one, CGG, it's a codon. Here you have UGA, which is another codon. And these, they're located in triplets. All right. And so this messenger RNA is going to help us. So what we would do is we take that codon chart that I had on the previous page, and we would go ahead and look at what AUG would fall, or where it would fall along the codon chart. Okay. So that's messenger RNA's role. It's going to be coding for the amino acid and the order of the amino acids to help us determine what our protein is. Now moving into the ribosome, and you can see the structure of the ribosome here up top. It is divided into a couple of different subunits. You have this A site and you have a P site, and in the bottom here we kind of tell you what each one is. The A site is, the P site is going to hold the growing polypeptide. So the growing protein molecule is going to be held at this location, okay? And then your A site is going to be the place where these new amino acids are going to enter that are attached to the tRNA. The tRNA, as I had just mentioned, is referred to as the anticodon, and because what it does is it actually attaches to the codon, which is your messenger RNA, but it's going to be the complementary pair to it that links up with it. So it carries amino acids to the ribosome. It's basically what it's doing is it's going out there finding the amino acid that it needs because it's been told by the messenger RNA, and it's going to bring that bring that amino acid in and eventually off into the ribosomes and start attaching to the polypeptide chain that is being formed. So you can see here with this anticodon, this is our transfer RNA here. What it's transferring is a certain amino acid that it has attached to. In this particular case, the anticodon sequence is GCC. So the messenger RNA, or the codon, would actually be here, would be CGG as we go along, and that would be what we would use in order to find what we're looking for on the codon chart. Okay, so the 3-base anticodon pairs with the messenger RNA, and it or what we call the codon. So your messenger RNA is the codon, your tRNA is the anticodon. They will eventually pair together, and that will form our polypeptide that we're looking to form with the protein. Okay, your amino acids. Now again, these are the building blocks of your structure. You're building blocks of proteins, and it's going to help us construct proteins. And depending on the order of them, depending on the length and the shape, basically determines what their function is. And every amino acid is going to have this general structure that you see here. It's always going to have this amino group, it's always going to have a carboxyl group, and it's always going to have an R group on top. Okay? And what you can see on the picture on the right here, these are two different amino acids. Notice the structures are the same here and here, and here and here. But then notice what they're going to do is when they bond together, as they tell you here, it actually has to lose the water in order for them to bond. And this bond that forms between them is what we refer to as a peptide bond. So when we put a bunch of these amino acids together, we form a polypeptide, which is basically a protein. So again, your amino acids are the building blocks of your proteins, and it puts together the structure, and that way it will tell us what the function is of that particular protein. Alright, so 3 steps of translation. Same 3 steps, just a little bit different. So what happens, we have initiation, elongation, and termination in the process of translation. And you can see in this little diagram here, you have transcription occurs. We're going from DNA up here to eventually into our messenger RNA. Now it's going to leave the cell through the cell membrane, goes off to the ribosomes, and then translation starts occurring. We're translating the language from nucleic acid language into amino acid language. Okay, so in the initiation step, as you can see in the animation, what's happening is the ribosomes are being constructed. You can see here this tRNA, right here, this transfer RNA is the anticodon. And then we have here this codon. This is referred to, AUG is referred to as a start codon because this tells us to start making the protein. And what it codes for, this AUG codes for methionine, or the amino acid here that starts the process. Okay, so you can a little bit of information here. You see the animation continuing. You see the rest of the ribosome being attached here into the P site, and this process of translation will begin here. And so, as I mentioned, you should know that a start codon is your AUG. That's the only start codon in our body, and it is going to attach to the anticodon, the transfer RNA, and be brought to the P site. So that's initiation. Okay, step 2, elongation. So this is where we're going to be adding in our new amino acid. And you can see the animation, the tRNA is being left, that was the methionine. There's a new tRNA that was brought in, valine, or the new amino acid. And then there's another amino acid coming into the A site. And then this one will move over to the P site. And then eventually we'll have, we would have another amino acid come in. Remember that this bond that's forming between the two amino acids are those peptide bonds. Okay, and that's going to attach the two amino acids together. So what we're doing is we're forming this polypeptide chain coming off in this direction, and we'll be bringing in the tRNA, right, that has went off and found the amino acid that needs to be attached here within the ribosome, okay, at the A site. And now the third step is termination. You can see again, transfer is leaving. It's already been attached. It's moving along the messenger RNA. We have attached and the new tRNA, and then you can see what's happening is this, what we refer to as a stop codon, comes in, and it actually released the new protein, the protein that's being made. So I'll kind of show you this here. Oops, let me go back here. Okay, so your stop codon has reached, and what we're doing is we're stopping the process. These are the 3 stop codons that are found within our cells. And remember, these are the messenger RNAs that are going to code for the anticodon to go off and find these stop codons, amino acids, and then it's going to stop the process of making the the protein. And then the protein is going to be cut off, and it's going to be released. Everything's released to go off and do what it needs to do next. Okay, so that's, again, that is translation. Now what I would like to do is go ahead and show you, or actually go through a couple of your diagrams real quick, and then I'd like to go ahead and talk a little bit, or show you a quick little video on that to recap this. So we're on page 91 in your packet. Okay, this is your transcription as it's noted here on top. And what we're doing here, this is step number 1. This is initiation. And what we're doing here, we have the RNA polymerase is a key factor here. There we go. And what that's going to be doing is it's starting to unwind it, and it helps add in, eventually add into the new nucleotides to the process. So you can see step 1 kind of carries over here. The 2 DNA strands are unwound and separates. And then what's happening here, the polymerase is going to add, as it says, the complementary nucleotides to this process. So this is really step 2, which is elongation. All right. And then eventually what we would have is step 3, which would be termination of the process. Okay, and that would be transcription occurring. Again, remember, this all occurs within the nucleus. We are going from DNA to our messenger RNA. All right, so that's that point. Now let's take a quick look at what translation does a little bit. This is a little bit smaller for you guys, but this is page 92, just the back side. Again, we're looking at translation. Remember, translation occurs at the ribosomes. Okay, and what we're doing is we're going from the messenger RNA to our protein, or what they refer to as the polypeptide. All right, so what we're doing and what you can kind of see is occurring here. Let me go ahead and zoom in real quick so we can see this a little bit better. Okay, so what's happening here, you can see this is our ribosome, this structure here. You can see the P site, the A site, and the new transfer RNA is coming in, and what you can see attached to that is the amino acid. All right, so this is going to be your initiation, step 1, and as these bonds form here, those are, as you can see here, your peptide bond, and that peptide bond forms between two amino acids. Again, remember your methionine here is your start codon. So it starts the process, step 1, and then we're going to move on to step 2 as where elongation occurs. Okay, so elongation is occurring. You can see the peptide chains getting longer. The transfer RNA is coming into the A site. Then older transfer RNA is going to the P site, and we're continuing making our protein. And eventually we get to the point of 3, which is termination. And this is the breaking and releasing, as you can see here, of all the different factors that are occurring. Okay, so what I'd like to do next is show you kind of a quick little video. It's going to be about 3 to 4 minutes long on the whole entire process of transcription and translation, and then we'll go from there.
The innermost workings of how a simple code is turned into flesh and blood. This is what Francis Crick called the central dogma of modern biology: how DNA makes protein. It starts with a bundle of factors assembling at the start of a gene. It's these that trigger the first phase of the process, reading off the information that will be needed to make the protein. The gene is the length of DNA stretching away to the left. Everything's ready to roll. 3, 2, 1. The blue molecule racing along the DNA is reading the gene. It's unzipping the double helix and copying one of the 2 strands. The yellow chain snaking out of the top is a copy of the genetic message, and it's made of a close chemical cousin of DNA called RNA. The building blocks to make the RNA enter through an intake hole. They are matched to the DNA, letter by letter, to make an exact copy of the A's, C's, G's, and T's of the gene. The only difference is that in the RNA copy, the letter T is replaced with a closely related nucleic acid known as U. You are watching this process, called transcription, in real time. It's happening right now in almost every cell in your body. When the RNA copy is complete, it snakes away from the nucleus and into the outer part of the cell. Then, in a dazzling display of choreography, all the components of another molecular machine lock together around the RNA to form a miniature factory called a ribosome. It translates the genetic information in the RNA into a string of amino acids that will become a protein. Special transfer molecules, the green triangles, bring each amino acid to the ribosome. The amino acids are the small red tips attached to the transfer molecules. There are different transfer molecules for each of the 20 amino acids. They all carry a specific 3-letter code that will be read by the machine. Now we come to the heart of the process. Inside the ribosome, the RNA is pulled through like a tape. The code for each amino acid is read off, 3 letters at a time, and matched to 3 corresponding letters on the transfer molecule. When the right transfer molecule plugs in, the amino acid it carries is added to the growing protein chain. Again, you are watching this in real time. And after a few seconds, the assembled protein starts to emerge from the ribosome. Ribosomes can make any kind of protein. It just depends what genetic message you feed in on the RNA. In this case, the end product is hemoglobin. The cells in our bone marrow churn out 100 trillion molecules of it per second.
Okay, so from here, what I would like to do, that was a good description kind of what's going on with translation and transcription. What I would like to do is the last thing is I would like to go through how to use this codon chart with you, so that way you'll be able to understand. I'm just going to do a quick example with you and go ahead and get it focused here for us. Okay, so we are on page 95 in your packet, and this is just going to look at the codon chart. Now there's a couple different setups for this codon chart, but this particular one, you can see the first base is along the left-hand side, the second base here, and the third base, and they kind of match up. So looking at this first one, let's go down to the bottom here, and what it's going to tell you here is you have the DNA. They want you to figure out what is the DNA sequence for this amino acid that you see is tryptophan. All right, so what we need to do is go up to the chart, find tryptophan, which you can see it is located here. All right, so we're looking at UGG. Now remember, the codon chart is coding for the messenger RNA. So you're going to go up to on the chart again, and what you're going to do is you're going to put UGG, and that is our messenger RNA. Now remember, if you have any T's in the RNA, you're wrong, because there only could be U's in that. So now what we're going to do is we're going to actually use that to create our DNA and to create our anticodon or our tRNA. So remember, this is codon here is your messenger RNA, this anticodon is your tRNA in the whole process of translation. So from UGG, what bonds with a U would be A, G would be C, and G would be C. And to kind of check yourself, we'll take a look at that here in a second, and then your anticodon would be U goes with A, G goes with C, and G goes with C. Okay, and what we can do is we can say, all right, UGG. So here's the first base on the chart. Here's U. The second base on the chart would be G, and the third base we said was G. So coming in here, it would be tryptophan. So we would be correct. Okay, and we'll continue on with these in class and do some more practice with using the chart for the sake of practice, so you get a better handle on what you're supposed to be doing with that. Okay, so kind of wrapping up the PowerPoint here on protein synthesis. Remember, we're moving from the transcription, which occurs inside the nucleus from messenger RNA, from DNA to messenger RNA, and then in translation outside the nucleus, we're going from messenger RNA to the idea of proteins. So this was screencast number 1 for the topic of proteins, and there'll be a couple more to follow on mutations and enzymes.